Before we discuss eDNA, the main topic of this video, I'm going to give a brief summary on what DNA is. Nucleic acids, lipids, proteins, and carbohydrates are the four major macromolecules essential for life. Amino acids are the basic building blocks of proteins. Similar to other organic compounds, they are made up of four key elements: carbon, oxygen, hydrogen, and nitrogen. Amino acids form into chains called polypeptides, and the amino acid sequence of a polypeptide is programmed by a unit of inheritance called a gene. Genes consist of DNA, which is a polymer belonging to a division of compounds known as nucleic acids. Nucleic acids are macromolecules that exist as polymers called polynucleotides, and each polynucleotide consists of monomers called nucleotides. Each nucleotide is comprised of three parts: a nitrogenous base, a pentose, and a phosphate group. There are two types of nucleic molecules: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. RNA is a more archaic and less stable version of DNA that evolved first. RNA chains are made up of ribonucleotides that are linked by phosphodiester bonds. A ribonucleotide in an RNA chain contains a phosphate group, ribose, and one of four nitrogenous bases. Structurally speaking, RNA is quite similar to DNA. Whereas DNA molecules are long and double-stranded, RNA molecules are shorter and single-stranded. RNA molecules consist of a single polynucleotide, whereas DNA molecules consist of two polynucleotides that spiral around an imaginary axis, forming a double helix. DNA also has a structural difference between the sugars, giving it more stability for storage of genetic information, whereas the instability of RNA results in it being used for short-term functions. Now we will look at the structure of DNA. DNA is made up of molecules called nucleotides, each nucleotide containing a sugar group, a phosphate group, and the nitrogen base. The four types of nitrogen base are thymine, cytosine, guanine, and adenine. The order of these bases determines the genetic code of the DNA, meaning the order of nitrogen bases in a DNA sequence can form genes that tell us how to make proteins. The aforementioned double helix structure resembles a ladder with sugar and phosphate molecules on either side, and the bases linking them. The bases on one strand pair with the bases on another. Adenine pairs with thymine, and guanine pairs with cytosine. In order to fit inside cells, DNA is organized into coil-shaped structures called chromosomes. Nucleus in cells also contain chromatin, which is a complex protein that condenses DNA molecules and prevents DNA damage during mitosis by controlling the splitting of chromosomes. Environmental DNA refers to DNA that can be extracted from environmental samples, such as soil, air, or water, without first isolating any target organisms. This DNA may be collected from a number of different substances, such as soil, snow, or seawater. Essentially, environmental DNA is mitochondrial DNA that has been released from an organism into its environment. Environmental DNA is formally described as genetic material obtained directly from environmental samples without any obvious signs of biological source material. Sources of eDNA include secreted feces, urine, mucus, gametes, shed skin, hair, and carcasses. It typically consists of a mixture of genomic DNA from several different organisms, containing the cellular DNA origination from living cells or organism and extracellular DNA. It is also possible for samples to contain some level of degradation, meaning DNA molecules that have become fragmented. The first reference to eDNA dates back to 1987, concerning a method of extraction of microbial DNA from sediments, where it was first utilized. In the 1990s, these same methods were used to monitor phytoplankton blooms and population changes in communities of bacteria. The usage of the term became more common in the early 2000s, predominantly in the microbiology community. In 2008, eDNA was first used to confirm the presence of an invasive species, Rana catespiana, in European ecosystems. In 2012, it was used to estimate the presence of aquatic vertebrates and the biomass of fish, namely the common carp. In the past 2 decades, eDNA research has progressed from the retrieval of small fragments of mitochondrial DNA to large-scale studies of ancient species and even whole mitochondrial genome sequences of extinct species. Environmental DNA has developed as an alternative method of sampling and species detection in recent years. Species detection is usually conducted using visual or acoustic direct observation methods. However, these traditional methods of observing species have several logistical shortcomings. On top of being expensive and time-consuming, many of these sampling methods, such as electrofishing and marine bottom trawls, are harmful to the environment. However, the emergence of eDNA has proven to overcome the aforementioned limitations of traditional sampling methods. Recently, eDNA has been harnessed to detect rare or invasive species and pathogens in a broad range of environments, and eDNA is improving accuracy, reducing costs, and streamlining workflows across many fields and industries, including paleontology, agriculture, public health, and ecology. In the context of conservation, eDNA analysis is already an essential tool in monitoring water quality, early detection of invasive species, and surveying endangered species. Environmental DNA is likely to be an area of development in the future, as it is being rapidly developed as a biomonitoring technique due to its high detection capacity, non-invasive nature, and rich taxonomic coverage. It is likely that future research will focus on the temporal and spatial distribution of eDNA in different habitats, and more precise links between eDNA concentration and species abundance. Future studies may also encompass the exact source of eDNA, whether from intestinal/epithelial cells, urine, or fecal matter, as these may vary depending on the lifestyle of an organism, thus affecting biomass estimates. Other areas of study will include chemical factors that affect eDNA, such as temperature, salinity, and pH. In future, it may be possible to obtain larger fragments than are currently targeted. This would be beneficial to the advancement of important conservation activities, such as population genetics using environmental samples and species-level identification. There are several key advantages to the usage of environmental DNA. One such advantage is that it allows you to sample from ancient environments. When applied to ancient sediment communities, it revealed DNA from several now extinct plants, birds, and mammals, giving us more insight into ancient flora and fauna. DNA from several invertebrates, namely insects, is shown to be preserved in ancient sediments, giving scientists an understanding of their evolutionary history. An advantage of being able to sample ancient environments is that you can see how they have changed over time and compare them with modern environments. Environmental DNA also plays an important part in conservation, as its use is becoming more common for species detection and monitoring. Studies based on eDNA have potential for applied conservation through detection of rare or endangered species, such as freshwater turtles. In future, it is likely that eDNA will be used extensively to survey turtle populations, as they are the most threatened group of vertebrates, with around half of their species considered of conservation concern. Due to the fact that rare species of turtle are hard to detect with traditional methods, detection using eDNA could be faster, more cost-effective, and more sensitive. Species that are of concern to conservationists due to low populations are usually the subject of eDNA sampling, such as the long-finned whale, the European eel, and eastern hellbenders. In conclusion, despite the aforementioned benefits of eDNA, there are still several pitfalls. Firstly, use of eDNA comes with the risk of contamination of samples taken, giving false results. Another disadvantage is the risk of inhibition, as humic acids that are extracted in DNA samples can corrupt the DNA, especially affecting water samples and thus affecting results. However, despite these shortcomings, eDNA is a good alternative method. Firstly, it is less destructive to the environment than other sampling methods, such as bottom trawling. On top of this, it doesn't require you to observe the organism in its environment, meaning that you do not need to go through the trouble of finding the exact location of the organism to observe it. In conclusion, eDNA is a new method of analyzing an environment that will continue to develop in coming years.
Thanks for watching. I hope you enjoyed the video. I spent several hours producing this video, researching, script writing, voiceovers and editing. All take a lot of time and effort. As such, if you want to support this channel, make sure to subscribe, like the video and comment any feedback in the comment section below. Also, share this video with anyone you think might be interested. More videos will be coming soon.